Assaf Vardi, who is a professor of plant and environmental sciences and Berkman professorial chair of plant science at Weizmann Institute of Science. He earned his bachelor's, master's, and PhD from Hebrew University of Jerusalem, covering biology, environmental sciences, and molecular ecology. After his postdoc research at École Normale Supérieure in Paris and Rutgers University, he joined the Weizmann Institute of Science faculty in 2010, where he studied molecular mechanism of microbial interactions in marine environment. Professor Vardi received a number of esteemed fellowship and awards, including Weizmann Institute Scientific Council Prize for Life Sciences in 2015, 2 Rieger Foundation awards for excellence in ecology and environmental sciences, the Marie Curie Fellowship, the Human Frontier Award, and many others. 20 years ago, he joined JGI for our first journey into algal genomes, the genome of Thalassia pseudomonada. And today, his talk will be about host viral dynamics in the ocean, a journey across scales.
Hello, everyone, and thanks for having me. Thanks for the kind opportunity to present our work from the last couple of years. Please email me for any question and comment about the talk. Your feedback is highly appreciated. I'll show a lot of unpublished data, so please do it. So the main theme of the work in my lab is to zoom in into the ocean and understand how microbial interaction can really shape the ecology and biochemical cycles in the ocean. We specifically zoom in to understand host virus dynamic, and we do it across scale, all the way by developing single-cell approach to study infection on the single-cell level, up to the plaque level, and even in induced blooms, as we will see in the fjords of Bergen, and even in the large scale of these blooms in the North Atlantic. The outstanding phenomena we studied for years is algal blooms in the ocean. As you can see here, although single-cell photosynthetic organism, they can form massive blooms in the ocean that can stretch over thousands of square kilometers. And that's important, hugely important, and is serving as half of the photosynthetic activity on Earth. We see this immense number of 50 billion tons of carbon a year. They're, of course, the foundation of the entire marine food web. And in the lab, we ask mechanistic question of how and what are the mechanisms that regulate bloom demise, because it's a huge source, a flux of organic carbon that fuel the ocean microbiome. So we will learn how different mortality agents, and specifically viruses, control algal blooms in the ocean. In one slide, the way we do it in the lab, we live in this tension between going out to the field and study this phenomena and interaction in algal blooms in a very complex environment, all the way to go back to the lab, bring isolates, developing robust model system in order to really go in depth into understand mechanism of pathogenicity, virulence, the molecule that mediate this interaction, and specificity. How does it matter when a cell is infected by a virus, by pathogenic bacteria, grazed by grazers, or how chemical communication within the population can shape the bloom succession. So let's focus on marine viruses, by far one of the hottest topics in the last decade in marine microbiology. And this is how it looks like when you take a drop of water from the ocean everywhere, anywhere, and you simply stain it with a DNA-sensitive fluorescent dye. You see this, the big guys are bacteria and the small ones are viruses that can reach up to 10 million viral particles in a drop of water. But a lot of important questions emerge because we still don't know how marine viruses can really shape the ecology of the ocean. That's what we study in the lab, especially the importance of the viral shunt, this ecosystem pathway coined more than 20 years ago, and suggested that the fixed carbon, we saw the immense carbon that is fixed in phytoplankton bloom, will be shunt towards product during the lysis of this bloom demise into the formation of massive flux of organic matter that then in turn can fuel the entire ocean microbiome and not being transferred to higher trophic level. So question we ask is, how do viruses impact the unique metabolic landscape in the ocean? How do viruses shape the structure and function of microbial lifestyle in the ocean? And what is the consequence of viral infection and bacterial consortia on large processes in the ocean? And the approaches we take to kind of demystify the concept of the viral shunt, or how we bring it back to the lab to study it in a more controlled manner, we developed over the years a very unique single-cell approach to quantify active viral infection. We developed a lot of platforms for environmental metabolomics in order to profile the unique metabolites and signaling molecules that are being produced during host-virus interaction. And also we generate a kind of synthetic communities from isolates from the bloom in order to mimic the dynamic, but really understand the mechanism and nature of communication. Very interestingly, unique to the marine environment are the giant viruses discovered more than a decade ago that can really infect a diverse host, as we can see here in this tree of life of eukaryotic cells. These giant viruses really change the old dogmas known for about, and really blur the difference between viruses and hosts. They can be hugely not just in size, but also in code to more than thousands of genes, really changing our view, the traditional view about passive virion and host. And the model system we use in the lab over the years to study host virus dynamic in the ocean, we studied the bloom, the massive blooms of Emiliania huxleyi. These single cell are not only beautiful and importantly, hugely important in photosynthesis, they also decorate by this beautiful exoskeleton made from chalk, from calcium carbonate. And we see these massive blooms that we can watch from space because they're so large. And even they're relict over geological time because of this exoskeleton that is released to the water upon infection and release into the water and accumulate in the ocean sediment. You see this beautiful example of the white cliff of Dover. So these blooms are not only hugely important for the carbon cycle, but also during bloom demise, they emit gas into the atmosphere. This is the smell of the sea that you all know. It's produced by a volatile organic sulfur called DMS, dimethyl sulfide, that only recently we discovered the enzyme that is responsible for the biosynthesis of this molecule that is hugely important, not just for the sulfur cycle, but also it emits to the atmosphere and can change cloud condensation nuclei. So very interesting to think how the fate of a single cell in the ocean can really change large-scale processes. Moreover, last year, Adva Shemi in my lab showed that this very important enzyme that produced DMS actually serve as a chemoattraction molecule for grazer that can really enhance their grazing upon production of this molecule by algal cells. Now, these massive blooms in the ocean can be infected by these giant viruses, Emiliania huxleyi virus. We see how this massive bloom here simply shrink over a few days by infection of these viruses. And these viruses encode in their genome up to 700 genes. And this is a large double-strand DNA virus. And it's encoded, and that's uniquely only to this virus to date in biology, for almost a full biosynthetic pathway for sphingolipid, a very important class of lipids. So over the years in the lab, we kind of changed the concept of just looking at viruses as passive particles in the ocean. And we are more interested to understand, in order to understand the impact on the environment, for this concept that we borrowed for Patrick Forter from Pasteur, that he called the infected cell the ViroCell. This is almost a new entity. And over the years, we invested a lot of work to understand the metabolic and transcriptomic changes that occur upon viral infection of the virus cell. So not any longer just looking at the particle outside the cell, but also to learn more about the virus cell life cycle. In order to understand host response to viral infection, viral strategy to take over, and eventually also develop sensitive biomarkers, both genes and metabolites, to quantify in nature the viral infection impact on algal blooms. As just one example, as I mentioned, the virus encode for, it's the only virus that encodes phosphingolipid biosynthetic pathway. So we took this as really important, really the heart of understanding the arm race between host and virus in this system by doing a dual transcriptomics. So we can quantify these genes of sphingolipid both from the viral side in the takeover of the virus and the shut-off of the host genes. And then we can also study how different are the genes involved in the biosynthetic pathway, host versus the viral encoded one. And we can also quantify by using lipidomics the unique lipids that are produced by each of these pathway, control versus virus cell infected cells. Lastly, we can use it because the virus is the end product of this biosynthetic pathway is production of this unique viral glycosphingolipids. We can use it as a sensitive biomarker in the blooms in order to detect viral infection in action. So we invested a lot of work to understand the remodeling of the viral cell lipidome, and here you by several approaches, one of which is comparative lipidomics, and we see a nice time course of infection, and this is the profile of the lipidome, and we see clearly how many new lipids are being formed during viral infection, and we can see which one and characterize them. The other approach we took is we combine classical plaque assay, that is, a classical method from virology from the 40s. We combine it with multi-imaging, multi-mass spectrometry in order to generate this beautiful metabolic map. So here we look at these two plaques here, and each map here is how the distribution, the spatial distribution of each molecules is being looked by through the lens of each metabolite. So quantifying this metabolic map can really help us now to understand the unique bioactive metabolites that are being produced during viral infection, all the way from the center of the plaque up to the periphery where the infection stops. And lastly, we're interested if there's such a profound remodeling of the metabolome during infection, what are the molecules not just that are being formed in the virus cell, but also some of them that are being released from the cell, and how it shapes the microenvironment of the cell. And Daniela Schatz in the lab, in a very, very important work, discovered that the infected cells are not only produce these giant viruses, but also produce extracellular vesicle, almost exosome-like, and in which she characterized over the years in the lab the unique cargo for these vesicles, and really found that they are discovered them as a new mode of cell-cell communication during infection. For example, Daniela discovered that these vesicles contain small RNA that can really shape the bystander cells, but also they contain important lipids that we see in the viral cell, but the vesicles encapsulate a very selective cargo. So it's not exactly as the virus cell, but only unique molecule will be encapsulated in the vesicle. And then we work very hard to understand what is the bioactivity of this vesicle. And we found that these vesicles can expedite in viral infection in the population, and also that they can extend the half-time of the infectious particle when we mix them, which is really important for a virus that is specialized on a specific host that appear in these blooms only once a year. It needs to survive in the water column with UV radiation throughout the time. And with an average half-time of 24 hours, this can be a very, very important issue for viruses for the next bloom to come. So new communication signal via extracellular vesicles that are being produced during viral infection. So one of the major efforts in the lab is kind of taking all the different infection states that are being produced during infection. We call it the ViroCell sphere, which is not just the virus cell at early stage or late stage. It's still a lytic virus, so some of the cell will induce programmed cell death. Some cell will survive, a minority of the population. And now we invest a lot of energy to understand the still unknown mechanism of resistance in this our system. And we do it among different approaches. And that's kind of transition in the lab over the years to move from understanding host-virus interaction in bulk, where we average all these interesting states on the population level, and move into understand individuality in host-virus interaction. So to understand cell-cell heterogeneity in response to viral infection in order to unravel new host response and viral strategy during interaction. And the way we do it is we develop and incorporate interesting single-cell approach to study host virus dynamic that gives us a powerful tool to quantify active viral infection, both in the lab and in the field, which is still a major bottleneck in order to understand the role of viral infection in the ecology. It will help us, of course, understand more about the enigmatic life cycle of giant viruses, and it will help also to map and match between the viral program and the host response on the single-cell resolution. And as I mentioned, we use it now to track rare subpopulation of resistant cells. Eventually, and that's very new project in the lab, we're also using single-cell dual transcriptomic approach in natural population in order to match and find the authentic host for specific giant viruses, which is still a big challenge in our field of aquatic virology. Three approaches we developed over the year for single-cell transcriptomic. First, we started with automatic microfluidics to capture the single cell and then do multiplexing qPCR on targeted specific genes. Then we adopted the MARSEC approach and 10x genomics to look at the whole transcriptome, dual transcriptome, both of the host and the virus on a single cell level. And also we had recently a very, very interesting approach to visualize active viral infection by detection of RNA molecule, again, the host and the virus, smFISH, single molecule fish. As I brought a few examples to demonstrate how important now is to increase the resolution for single cell approaches. Here we see, for example, from our work, each dot here is a single cell. We're looking now at the viral transcriptome. These cells are not induced at the viral program, where these cells are strongly inducing viral gene expression. So already by this one view, one time point, we see this very interesting heterogeneity and individuality of these cells in this population that is completely identical genetically. And that gives us a powerful tool to really start to ask questions why these cells are highly infected and these cells not. If we simply continue the usual average bulk RNA-Seq, this is how it will look like, this one average of everything. And thus we use this heterogeneity now. And what we did here is simply reordering bioinformatically the cells based on their transcriptional program, all the way from early state of infection to late. This gives us now a powerful tool now to look at new resolution at viral infection. And we don't care anymore if it's 2 hours or 4 hours or 24 hours post-infection. We can capture the infection and define the infection state, which means every row here is a gene, a viral gene, and every column is a single cell. So by reordering the cells now and transcript based on the gradient of infection state, we start to see and unravel this very interesting viral transcriptional program. That when we look now into the gene, we can really start to define clusters of viral genes that we help us to define them in different kinetic class, immediate, early, and late. Very interestingly, that enable us to discover that they are shared in the promoter region, a very interesting motifs that each cells in specific and each genes in a specific kinetic class share this motif, which was very interesting finding for us. So let's move on to another approach of single cell to visualize single cell resolution, both virus and host transcript, and it's called single-cell FISH. And that can enable us, for example, to watch and learn on a subcellular level about the viral life cycle. It can help us now to also to quantify in a high throughput manner, because we can use flow cytometry and image stream. And also it can enable us to look in a mixed population in nature on specific pairs we want to study. That's very, very important when you look and study a very complex environmental samples. And this is how we look like. Here we probe both a host gene, a photosynthetic genes as kind of proxy for the metabolic state of the cell, and MCP, major capsid protein, a viral gene. So we look at RNA, both of the host and of the late viral gene. At early state of infection, of course, a strong photosynthetic gene expression. But late in the infection, we see this beautiful image here with shining dots from viral strongly gene expression, and in parallel, a nice shutoff in the host gene expression. This is in microscopy, but we can take it to flow cytometry, and we use it now routinely in the lab, really in a high throughput manner. We see this nice time course where each sample now we can analyze thousands of cells and learn about their infection state. Starting here with the major capsid viral protein, we see the gene expression and nicely how it's being proceed over time. So we can really quantify the fraction of active viral infection. And in parallel, we see this nice shutoff of the host photosynthetic machinery. This is just quantification of these flow cytometry plots. It really helped us now to discover that in early state of infection, there are viruses being produced without lysis. So we discovered this non-lytic viral infection supported by this electron microscopy work that Daniela did in the lab, and to really see that the cells stay intact but still produce these giant viruses. And from the other end, the lysis that we typically see within a couple of days comes much, much later. So if we stick with, this is just a beautiful example in which when you look into the virus cell fate, you can start to decouple between the infection state and the fate of the host cell. Where if you keep looking at host virus dynamic on extracellular, looking at viruses going up and hosts going down, and a kind of typical predator-prey interaction, you will miss this interesting life cycle that takes place intracellular in the virus cell. Nir Yofi in the lab, that finishes master, really beautifully described a new state of the host and the virus. We typically study what happened here in the lytic phase. We see here in the first 3 to 4 days, a very sharp decline in the host due to the lytic phase of the virus as compared to control, and this is viral production. But with the new study, Nir identified that only a rare subpopulation can always recovered and got into a stable coexistence of hosts that is resilient to viral infection, and he tried different Emigliani-Huxley virus. And even more interestingly, that the viruses here keep being produced. So we have a population of the host that recover. It's completely resistant to external addition of new viruses, but it still produces viruses in a persistent manner. Very interestingly, when Nir applied the single-cell FISH approach, he identified that actually we're looking at a very interesting phenotypic heterogeneity, where majority of the population does not contain viral transcript, but around 5% of the population is actively infected. So we have very interesting balance here between these two subpopulations and how it's being maintained over, and how the stability between resistant strain subpopulation and highly infected, and what are the molecules that now mediate this interaction. This is a major focus in the lab. So let's shift now into the complex marine environment, where we don't look like in the lab just on one virus and one host, and how with the new approaches we bring in, how we quantify viral infection in natural blooms, and also what is the unique metabolic landscape that is being produced during viral infection. And to do so, we induce the bloom here in the beautiful fjords close to Bergen, and we see this very strong Emigliani-Huxley bloom being introduced. Simply, we're taking these 11 cube bags that are immersed in the water, and we simply use inoculum, natural inoculum from the fjord, and we just add water. And that's really, over time, we see, and we did it in several bags, we see very interesting dynamic, very strong bloom formation, succession of Emigliani-Huxley, reaching close to 100,000 cells per ml, and a strong demise, a strong crash of the bloom, and viral production. Very interestingly, we see that although the bloom almost in a synchronized manner demised, the level of viral production is very different between different bags. So that gives us a powerful tool to really dissect and disentangle the specific role of viral infection in, for example, metabolic production, and how it shapes the unique metabolic footprint in the environment. We also applied some of the single-cell dual transcriptomic approach I showed you in the lab, and Gur Hebroni, a former postdoc in the lab, did it for the first time in natural population over thousands of cells. And here we just see that it gives us a powerful tool. Each dot here is a cell and level of viral expression within each of these cells. So we can really start to learn and understand the pandemic of viral infection in the ocean using this powerful tool. Using a good single-cell approach, we can have really ultra-sensitive approach to detect viral infection at very early state of the bloom, way before the bloom arriving the demise phase, which really help us now to try and decipher between the fate of the individual cell and the fate of the bloom as a population. Again, with the excellent work of a former postdoc in the lab, Flora Vincent, that developed the entire single molecule fish that we saw before, she did the same for natural population, this time taking the complexity and really zoom into understanding the dynamic of hosts of Emiliania huxleyi and its specific virus. This is the extracellular production and the bloom demise. And we start to visualize here the fraction of infected cells. And there came the big surprise. Although the bloom crashed in a synchronized manner just within a few days, and we see viral production, when we look intracellular on the fraction of infected cells, it reached only up to 30% of the population. So how come the entire population died together in a synchronized manner? That really opened our mind to a different mortality agent that might act in concert with viral infection. It can be, for example, metabolites that we study in great depth that are being produced from the infected cells and vesicles, as we showed before. It can be a co-infection together with grazers and parasite, but it wasn't the case here. It can be also high turnover of the infected cells, and eventually, and that's emerging field of research in the lab in the last few years, is how viral infection can act together with pathogenic bacteria that also can kill the algal cells. So in a way, how these two top-down regulator can control the fate in the bloom. And indeed, we see that in this phase of viral production, with a small shift in time, we see production of bacteria. And from this phase, we have now a nice repertoire of bacterial isolates that we assay in the lab, and some of them, especially from the group of Rosobacter that usually associate with these algal blooms, can be pathogenic to bacteria, and we study this mechanism. So do viruses and pathogenic bacteria can act in concert during bloom demise? You know, akin to what happened in our body, for example, where viral infection, influenza, for example, is going first, and then a second invader of pathogenic bacteria like pneumonia, and can utilize some of the metabolites and how they act in synergy to kill host cells. But it was never studied in the context of algal blooms or in the marine environment. And for this aspect, we now archived microbiome, we call it BAMs, bloom-associated microbiome, from different blooms that we study over the years. And then we develop now synthetic communities in the lab that are composed by this tripartite interaction between the algae, the specific virus, and the associated bacteria. And we start to learn the unique metabolic exchange that are being produced and how synergy is acting. What is the mechanism? So how by this approach, we can move from the ocean back to the lab in order to understand more mechanistically about this interaction. And indeed, Eliran Sofer and Noah Barak in the last recently in the lab did fantastic work really to show this synergy. We see that here one example in which we take bloom-associated microbiome from the bloom demise, and we added to the algae together with the virus in the lab, also as isolated from the bloom. And we see this very interesting synergy in cell death of the algal as compared to the control just with the virus. Also, when we add antibiotics, we see that it's getting very close to virus only, meaning there's a very interesting metabolic crosstalk, which we can then also recapitulate by isolating from this microbiome specific bacterial, pathogenic bacteria that can do and have the same bioactivity. So we call it partners in crime, how viral infection now primes bacterial pathogenicity and how they can together work in synergy during algal bloom demise, completely new concept in marine microbiology. And lastly, Connie, an excellent postdoc in the lab, together with Guy, a very talented postdoc in the lab that left the group more than a year now, studied the unique exometabolome that is being produced during viral infection. And the way they did it is by really establish a robust exometabolome platform, environmental metabolomics, so we can take samples from the environment and really start to assess. And they did it about the molecules that are being produced and released into the environment and really shape now the bacterial lifestyle. So what we can start see here, this is profile of close to a few thousand of metabolites or mass feature, as we call it. And we can cluster them and then really start to define unique metabolites that are unique to different phases of the bloom. And when we zoom it in further, and here is this nice four blooms, the host dynamic, and they're different. Only in bag 4, we see very strong viral infection. So we can look and compare between their exometabolome and look for unique metabolites that are unique to viral infection as compared to other bloom that did not die from viral infection. And also we use correlation to viral gene marker in also really to distill and extract the unique metabolites for viral infection. And here Connie and Guy really identify unique set of molecules, chloroiodo metabolites that are very unique and rare in this combination that are strongly induced during this unique metabolic footprint of viral infection. We can then use this metabolic biomarker, not just in a mesocosm sample, but also in the real world in massive blooms of Emiliania huxleyi in the North Atlantic. And also they founded there these unique halometabolites, halogenated metabolites that are being formed in the late infection state of the bloom in the North Atlantic. So viral infection leaves unique halogenated footprint in oceanic blooms. And now we are learning in great depth what is the role of these molecules, of these halogenated molecules that are also very unique to the marine environment, in order to understand what are the enzymes that generate them, what is like halogenases, for example, and what is the bioactivity, because in many cases, and that's from the natural product chemistry world, many of these halogenated molecules can act as antiviral and antibacterial molecules. So we're extremely interested to understand, together with a cool collaboration with Rebecca Goss, to understand about the origin of this molecule and their function. Very interestingly, we saw this strong metabolic biomarker footprint in the ocean, but how do we bring it to study it in the lab? So and that's Connie, with the major effort in the lab, generate these synthetic communities, again, made by this tripartite interaction, algal isolates, together with viral isolates from the bloom, and a unique microbial and bacterial consortium that associate and isolate together with the algae. And what we see that only in the combination of these tripartite, we see production of these halogenated molecules in the lab. So now it gives us a powerful tool to isolate the enzyme and really understand whether it's the viral infection responsible for the chlorination as the first phase and bacteria as the second phase of iodination. So really understand the dynamic of this tripartite interaction that we are very keen to really understand and isolate this molecule and understand their function. Lastly, this is the last slide. We want to understand how this microscale interaction that we study in great depth in the lab and in the field, how they will shape the carbon cycling in the ocean. From one hand, whether they will just enhance the viral shunt, recycling of nutrient in the ocean, or that they will enhance carbon export into the ocean, because we found previously that viral infection really enhance polysaccharide production, which makes the cell sticky and aggregate, and then can enhance the efficiency of export to the deep ocean. So very interestingly, how this microscale interaction between the tripartite will impact large-scale process as the carbon cycle in the ocean, and that's a major now challenge for us to better understand. Most importantly, for me to understand the great team of mine in Weizmann, I'm so fortunate to have this very, very talented present and former student that contribute to the amazing work in the lab, our collaborators, and the team we work with in Bergen to induce the bloom and in other field studies. And please, again, send me any comment or any question about the talk. This is my email. And interestingly, if you are interested in any of this subject, we always look for postdoc and talented students to join the lab. So please come visit. You're more than welcome. And to join this amazing group of people that we know also how to have a great fun in the lab. So thank you so much.